understanding the variants of SARS-CoV-2. We want to understand how well vaccines will protect against infection with the variants, and we want to understand why these variants are spreading with increased prevalence. So in this study, we were particularly interested in the Lambda variant. This is a variant that really has taken off in South America, particularly in Peru, where it went from initially low prevalence to now being the major virus present in that country, and is spreading now throughout South America, and it has been found in many other countries, including the United States. The variant has mutations in the receptor binding domain of the spike protein, so that's the part that contacts the receptor ACE2. There are two mutations in particular. One is at position 452, the other is at position 490. In addition, in the amino terminal part of the spike protein, there's a 7 amino acid deletion. So in order to look at that variant, we generate what are called pseudoviruses in the lab. These are based on HIV virions, but they have the spike protein of whatever SARS-CoV-2 variant we would like. So we made the Lambda pseudoviruses, and we tested them with sera from people who had been previously infected with the original virus, or who had been vaccinated with any of the 3 approved vaccines here in the US. What we found, and we were quite pleased to see, was that the mRNA vaccines, although they are lower in their neutralizing antibody titer against the Lambda variant, they still result in good, or titers that we believe will result in good protection against the variants. The J&J vaccine had somewhat lower titers than did the Moderna and the Pfizer mRNA vaccines. In addition, if we looked at the infectivity of the virus, we could see that, in fact, those variant viruses, so the Lambda pseudovirus, was more infectious than the wild-type virus, and this was due to position 452. So the virus is being selected by an evolutionary process that allows the virus to become more transmissible. What's fascinating is to see the virus evolving in real time. So these small changes, so single amino acid changes in the receptor binding domain of the spike protein, allow the virus to become more infectious, as well as to escape neutralizing antibodies. So we can see this virus evolving in real time. So that one single amino acid, position 452, allows the virus to bind more tightly to ACE2, and it also allows the virus to escape the neutralizing antibodies. So we see the evolutionary pressure on the virus and how it responds by mutations. So after we did this study, we did a very similar study with the mu variant. And what we found was that that variant was neutralized quite well by the vaccine sera, and did not have increased transmissibility or increased affinity for the receptor. So we don't believe that that variant is going to pose any new problems over what there was already. Interestingly, in that same study where we looked at mu, we looked at another variant, a variant that's not very prevalent, it's quite rare, but it has quite a few mutations. That virus now has just been called C.1.2. And what we found was that virus really is quite resistant to the vaccine sera. And we were concerned about that because the concern is really that a variant might pop up that is quite resistant to neutralizing antibodies. And this virus is quite resistant. But what's interesting about it, it has lower affinity for ACE2. That is, its transmissibility is going to be less than the earlier viruses. So the virus tried to escape from neutralizing antibody, but in doing so, it becomes less fit. And we think that's a good thing, because while new variants may emerge, they are likely to be less fit and therefore will not take off and spread rapidly throughout the human population. Well, there's really two limitations that we should consider. One is that we have only looked at neutralizing antibodies. It's important to keep in mind that antibodies have other functions in addition to neutralization. They also have effector functions. For example, when they bind to the spike protein of a virus, they allow the virus to be phagocytosed by cells such as macrophages. So that's another way that antibodies work in vivo, although we have not measured any such effector functions in our experiments so far. The second thing to consider is that we have not looked at the T cell response against the variants. So I think what needs to be done next is to really understand what are called the correlates of protection. So we, in our studies, we look at the neutralizing antibody titers, but it's hard for us to say in reality, how much does that protect a person from being infected? So when we measure, say, a titer of 1 to 100, what degree of protection against infection does that provide? That needs to be studied by doing large clinical trials of measuring antibody titers in people and then testing the probability with which they will be infected over time.